Question number 1. The 35 sequence. Question number 1. The 35 sequence is for the 6 base pair region recognizable in most promoter. This hexamer is typically 35 base upstream from the start site. The cosinus 35 sequence is. Answer is option C, TTGACA. Question number 2. The most conserved sequence in sigma 70 promoter is a, which is found in the promoter of many different E. coli gene. 30 base pair sequence, 20 base pair sequence, 6 base pair sequence, 12 base pair sequence. Answer is option C, 6 base pair sequence. Question number 3. The initial unwinding of DNA in transcription process result in formation of an open complex with the polymerase, and this process is referred to as holoenzyme, tight binding, close complex, coenzyme. Answer is option B, tight binding. Question number 4. The RNA polymerase remains bound to the DNA and continues transcription unit. It reaches a dash at the end of transcription unit. Promoter sequence, stop signal, terminator sequence, B and C are correct. Answer is option D, B and C are correct. Stop signal and terminator sequence. Question number 5. The RNA hairpin is often followed by a sequence of 4 or more at dash residue A, U, C, G. Answer is option B, U, uracil. Question number 6. The TRP operon encodes dash structural gene involved in tryptophan biosynthesis 3, 5, 6, 4. Answer is option B, 5 gene. Question number 7. Tryptophan, the end product of enzyme encoded by TRP operon, therefore acts as a co-repressor and inhibits its own synthesis through CRP protein, TRP promoter sequence, end product inhibition. All of the above are correct. Answer is option C, end product inhibition. Question number 8. The most common stop signal in transcription process is dash in which the RNA transcript is self-complementary. RNA hairpin, transcription bubble, tight binding, open complex. Answer is option A, RNA hairpin. Question number 9. The dash is responsible for recognition of co-sequence promoter sequence and is only required for transcription initiation. Attenuator, sigma factor, beta subunit, alpha subunit. Answer is option B, sigma factor. Question number 10. The dash subunit is most common sigma factor in E. coli, which is responsible for recognition of general promoter which have cosinus 35 to and 10 elements. Sigma 35, sigma 50, sigma 28, sigma 70. Answer is option D, sigma 70. Question number 11. RNA polymerase 1 is located in the nucleus, responsible for synthesis of the precursor of most rRNA, responsible for the synthesis of mRNA precursor and some small nuclear RNA. A and B are correct. Answer is option D, A and B are correct. Question number 12. In eukaryotes, the pre-rRNA transcription units contain 3 sequences that encode the 60S, 5.8S and 20S rRNA, 80S, 5.8S and 20S rRNA, 80S, 5.8S, 80S rRNA. All of the above are false. Answer is option B. Question number 30. Pre-rRNA is synthesized by dash in the nucleus. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3 primase. Answer is RNA polymerase 1. Question number 40. Selectively factor 1 SL1 is made up of 4 subunits. These include the Tata binding protein, which is required for transcription initiation by all 3 RNA polymerases. The other factors are RNA polymerase 1 specific TBP associated factor called TAFIS, TTGACA, RTBP, SL1 TBP. Answer is option A, TAFIS. Some eu Question number 50. Some eukaryotic genes contain a dash instead of a Tata box located around the transcriptional site set. Enhancer SPL box, initiator element CCAAT box. Answer is option C, initiator element. Question number 60. RNA polymerase 2 is located in the nucleoplasm, located in the nucleoli, is responsible for the synthesis of mRNA precursor and some small nuclear RNA. A and C are correct. Answer is option D. A and C are correct. Question number 70. Which of the following is true for RNA polymerase 3 located in nucleoli? Responsible for the synthesis of precursor of 5S RNA, tRNA and other small nuclear and cytosolic RNA. Responsible for the synthesis of mRNA precursor and some small nuclear RNA. Responsible for the synthesis of the precursor of most rRNA. Answer is option B. Question number 80. The enhancer have the following general characteristics. They exert strong activation of transcription of a linked gene from the correct start site. They activate transcription when placed in either orientation with respect to linked gene. They are able to function over long distance of more than 1 kb whether from an upstream or downstream position relative to the start site. All the above are correct. Answer is option D. All the above are correct. Question number 90. Ribosomes are complex of dash molecules and specific ribosomal protein tRNA, rRNA, mRNA, RNA polymerase 3. Correct answer is option B, rRNA. Question number 20. In the prokaryotes E. coli, there are 7 different operons for rRNA that are dispersed throughout the genome. Each operon contains one copy of each of the. Answer is option B, 60S, 60S, 5S and 23S rRNA sequence.